Hello students, let us continue our chapter digestive system, and today we'll be talking about certain experiments on digestion. The first one is action of saliva on starch. So here what we have to do, we have to take three test tubes. In test tube A we have taken starch, in test tube B we have taken sugar, in test tube C we have taken starch plus saliva. As we know, saliva contain an enzyme, that is salivary amylase, that helps in the breakdown of starch to give maltose. And also we know the test for starch, that is when we add iodine to the starch solution, it turns to blue black in color. So if we pour iodine to this solution, we'll find that the solution will turn to blue black in color. In sugar, we'll not find any change. In test tube C, if we'll do this, as if we'll perform the iodine test, we'll find that it will come negative. It will show negative test because as the starch is present along with the saliva, so saliva will act upon starch and will convert it into maltose. And also will heat the solution to 38 degrees Celsius, that is the body temperature, so that the enzyme can easily act upon the starch. So this was the action of saliva on starch. Means if saliva is present, then the starch will be converted into maltose. Then coming to the action of pepsin on egg white. Here we have taken 4 test tubes, test tube A, test tube B, test tube C and test tube D. In each test tube we have taken egg white. Egg white contains protein. So in test tube A we have taken egg white along with pepsin. In test tube B we have taken egg white along with HCl. In test tube C we have taken egg white along with pepsin plus HCl. In test tube D we have taken egg white along with acid, that is HCl plus boiled pepsin solution. We'll leave it for few minutes. We'll heat the solution for a while, and then we'll find that we'll test for protein. We'll find that test tube C will show the change in color, means the clear substance will change into cloudy suspension, while rest 3 test tubes won't show any changes. That means pepsin cannot act without HCl. HCl cannot act upon egg white without pepsin. Egg white cannot be acted upon by the acid along with the boiled pepsin solution, because if we boil the pepsin, enzyme will be destroyed. But in test tube C we have taken pepsin plus HCl, and this solution will give a favorable condition for the enzyme to act upon the egg white. Now we'll be seeing the same thing, but we'll add one test tube, that is to see the effect of alkalinity on the enzyme reaction. So the same setup we have taken A, B, C, D along with one more test tube in which we have taken pepsin plus sodium bicarbonate. So sodium bicarbonate is alkaline in nature. So in this case also we'll see the change occurring in test tube C. Why? Because in test tube E we have taken pepsin along with alkaline solution that will not perform, or that will not allow the enzyme to perform the reaction. So in this case we prove that the acidic solution of pepsin, as well as in a particular temperature, it will act upon the protein and it will convert it into peptide. Now we'll be going through some of the tests, that is glucose test for glucose. So as we know, glucose, if we'll add some ferring solution to it, first the solution will turn to blue and then to green, then to yellow and orange, and lastly it will turn to deep brick red in color. Then there is a test for starch, in which we'll take starch plus iodine. If we'll add iodine to the starch, it will turn to blue black in color. Then the test comes for protein, where if we'll take egg white along with nitric acid, we'll find there will be a color change, that is white to yellow. Then we'll take the same solution, we'll add some water, we'll rinse the acidic solution from that, and then we'll add some ammonium hydroxide, that is alkaline in nature, and if we'll heat it, then we'll find that the yellow color will be changing to orange in color. So we can uh, prove that the solution contains protein. Then comes test for fats and oils, that we can perform it anywhere, like we can take a piece of butter or walnut and we can rub it on a piece of paper, then the spot rubbed turns translucent, especially when viewed against light. So these all were the tests for on food, and hence we can prove that the substance that is contained in that particular type of food. So we have completed our chapter digestive system.